All right, everyone's in. All right, everyone. Thanks for taking the time and joining us tonight. Shout out to any ESF Herpetology Club members who were present. We appreciate you guys co-hosting this event with us. Um, Tonight we are grateful to be joined by Dr. Donny Walker, ESF alumnus and associate professor of biology at Middle Tennessee State University. Tonight, he will be treating us to a discussion on some of his recent work with fungal pathogens associated with the snakeskin microbiome. Thank you, Dr. Walker, for taking the time to share your work with us tonight, and feel free to get things started whenever you're ready.
Awesome. Thank you, Ross. Yeah, and thank you all for attending. And yeah, I have a warm spot in my heart for ESF, BSF campus, Cranberry Lake, and yeah, it was just a wonderful time. I graduated in 2007 from ESF, so it's been a minute since I've been back to Syracuse. But yeah, I'll talk to you a little bit about my research, the program that I've built over the last about 9 years working on snake fungal disease and snake skin microbiomes. And so I know it sounds kind of like a relatively complex title, but really what we're looking at is how the snake fungal disease pathogen causes dysbiosis or like disruption, like if you think of like disturbance ecology, how it might cause a disturbance to a skin microbiome as a result of pathogenesis. So if you're interested in seeing kind of like what we do in the laboratory, I have my QR code for the lab webpage here. And again, at the end of the talk today. But before we get started, let's see if I can hide this. Yeah, before we get started, I think it's important to give credit to the individuals who have contributed to this work. It's mostly me getting to take credit and talk about all the hard work that my graduate and undergraduate students and postdocs have done over the years. And so huge contributors to this work include Dr. Matt Grisnick, Alex Romer, and some undergrads, Jackson and Emma here. Kylie Moe here transitioned from undergrad into being my lab manager. And then more recently, I've hired on Dr. Jason Dallas as a postdoctoral researcher to focus on snake fungal disease, as well as Dr. Caitlin Murphy and Dr. Mitra Gotby in the front here to focus and work a little bit more on this project. And so we've also been really lucky enough and fortunate enough, I should say, to have recently been awarded an NSF career grant to continue studying snake fungal disease and dysbiosis to the microbiome. And so that's some of what I'll talk to you about today. And I think it's really important to keep in mind when we discuss microbiomes, we need to think that it's like not just the bacteria that make up the microbiome, but it's rather the bacteria, the fungi, protists, and the viruses that are all interacting with one another to form these microbiomes. And these microbiomes are really important, and bacterial fungal interactions are really important in structuring individual host health, populations, communities on the landscape, and overall ecosystem function. And so before transitioning into a bunch of data heavy stuff, I thought I'd first kind of give a shout out to all of our microbiome donors that we study in the laboratory. These are all species of conservation concern in the state of Tennessee, including the green salamander, the four-toed salamander, the streamside salamander, which may soon be federally listed, unfortunately, the timber rattlesnake, the pygmy rattlesnake, which is really cute and tiny as the name suggests. And I think it's important to just like think about how these organisms, these hosts as we call them, interact within an environment to form these communities. But really what I'm most interested in when thinking about hosts and their microbiomes is this. Like I love to get out on the landscape and I love to chase these creatures around. It's a lot of fun. And sample them. But really what I'm most interested in and what gets me most excited is a Petri plate that looks like this, where we see all sorts of really cool and interesting bacterial fungal interactions. So for example, here's a bacterium producing some form of antibiosis. You can see that and notice that by the zone of inhibition. You can see a fungus over here, perhaps being partly competitively excluded by another bacterial species, but not completely. There's all sorts of interesting communication between the bacteria and the fungi on this Petri plate. And this is really what drives me back to the lab and the office on a day-to-day basis. So I thought I'd give you kind of a bit of a glimpse into what we do in the laboratories. We do a lot of field work, as you can see in some of these pictures. And, pictures only kind of partly do things justice. And so I thought I'd pull up a quick video of some of we have a bunch of lab videos from the field, from all of the different microbiomes that we've studied, but this one is particular to snakes and snake fungal disease. You'll have to let me know, Ross. Can you hear this? Yeah, cool. Okay, so we strap down a bunch of gear and a truck, and then we do something that you herpetologists know called road cruising. So we drive for hours and hours and hours. Sometimes we lose our gear until we find a snake. The snakes are oftentimes venomous, and so we use a pair of tongs to slip a plastic tube over the head and neck of the snake. And so after that, we rinse all of the transient microorganisms off of the snake. We examine the snake with a flashlight and for clinical signs, and sample its microbiome using a standardized protocol. So we do a fair amount of field work. We also switch this around again. There we go. We also use quantitative PCR to determine the presence or absence of the snake fungal disease pathogen, as well as quantify fungal load. So you're going to hear me talking a bunch about fungal load throughout this talk, and these are data that are generated using quantitative PCR. And you can also see some of the clinical signs here associated with snake fungal disease, from really subtle on the left-hand side to pretty extreme, like this animal is probably blind and is going to be picked off by a predator in the very near future. Another methodology that we commonly use in the laboratory is metabarcoding and high throughput amplicon sequencing. And so this allows for us to take the skin swabs collected from the snakes, do total DNA extraction on it, and use some barcoding markers for both bacteria and fungi. We generate millions and millions of DNA sequences that then run through bioinformatics processing to identify particular species and what they might be doing in a snake skin microbiome. This really allows for us to, as the slide says, kind of like study the structure of the microbiome. Another one of the staple methodologies that we use in the laboratory are these in vitro microbiological assays to determine how microorganisms might respond to a particular carbon source that mimics the snake skin chemistry. And so I'll show you some results associated with this in just a bit. We do a handful of live animal inoculation studies to understand how the fungus actually affects the host. And we do all of this because emerging infectious diseases are quite important. As you might know, emerging infectious diseases are increasing in the 21st century. Good example of this is the chytrid pathogen, which has caused extinctions and extirpations of nearly 500 different species of amphibians. As this graph indicates, this is unfortunately happening worldwide, and fungi really represent a disproportionate percentage of disease-causing organisms. And so we do this to understand and really take into account these host microbiome pathogen interactions. And so this is kind of an outdated figure, but I really like it because it's simple. It's called the disease triangle. It indicates to us that a virulent pathogen, a susceptible host, and a favorable environment are all required for disease to manifest. And when thinking about host susceptibility, we take into account, or should take into account, both adaptive as well as innate immunity. And we as a lab group study the host skin microbiome, which is really an extension of host innate immunity. So what I'd like to do now is kind of transition into telling you the story about fungal pathogen-induced dysbiosis to snakeskin microbiomes. And once again, this is just highlighting some of the students. Dr. Cody Godwin, who's now an assistant professor at Santa Fe College, Dr. Matt Grisnick, who's now an assistant professor at Coastal Carolina University, Jake Leese, who completed his master's degree, and Alex Romer, who also completed his master's degree. Both went on to PhDs as well. And a whole long list of undergraduate students that I can't even really fit onto the slide. There have been a lot of undergrads who have contributed to this work over the years. Okay, and so as you probably know, especially if you're a herpetologist, or I guess maybe if you've looked for herps in general, you know they're cryptic. They're really hard to find, snakes especially. And so to study snake fungal disease in Tennessee, we established the Tennessee Snake Fungal Disease Working Group in 2015. This has since grown to 40 participants in 30 different universities and governmental institutions. We've now done, this slide is a bit outdated, 9 years of surveys. We're just starting the 10th year of surveying this year. We've collected probably well over 1,500 samples at this point. My lab group at MTSU has largely led the coordination and processing efforts. However, I've had a lot of help from a colleague up at Tennessee State University named Bill Sutton. He is a real herpetologist, and Chris Murray down at Southeastern Louisiana, who is also a herpetologist and crocodilian biologist, who have helped lead these efforts. And we've done this to study the fungal pathogen Ophidiomyces, ophidiicola. So this is the snake fungal disease causing pathogen. It causes cutaneous infection that can spread to more visceral and internal organs. It's associated with increased skin shedding, lethargy, and death. And this fungus is really, like its life history, geographic distribution, host distribution is really poorly understood. I'd say it's really only been studied for maybe about the last 13 years or so. It's kind of popped up onto the map of infectious organisms that we should probably be aware of. And so my lab as a whole places a strong emphasis on understanding the importance of scale in ecology, because as community ecologists, we are interested in if the communities we observe are true and accurate, or just random communities captured at a certain time and place. So for example, if we look to the map here, you can reveal fine scale mechanistic details if you make observations at fine scales. However, generalizations are more likely to emerge as we scale out to broader scales. And so when we're designing experiments, we really emphasize the importance of making measurements across experimental scales to more accurately reflect true system processes. And so the way that we approach this is we scale from ecosystems to synthetic microbiomes. We do studies on the landscape where we have basically low to no experimental control. We do live animal controlled studies where we have moderate experimental control. However, can't really control everything, as we're oftentimes bringing these animals in from the landscape. And then we do synthetic microbiome studies where we have complete control, very high experimental control. We can mimic everything inside of a 96 well plate. For example, we can select particular compounds that mimic snakeskin chemistry to understand how bacteria and fungi respond to those carbon sources. And so I'll tell you and show you examples of experiments from each of these different scales today. Right, and so the first and most basic question, and really a major assumption, is like, do snakes actually have a skin microbiome? And so this ordination is showing you that, yes, indeed, snakes have a skin microbiome. The pink samples here all cluster together, and they are distinct from the environmental samples that we've collected. However, you can see in the Venn diagram on the right that there is indeed some overlap between the snakeskin and environmental samples, and these are bacterial microbiomes. We refer to them as OTUs, or operational taxonomic units. They're basically like the molecular marker that we've sequenced. It's a molecular sequence that can be mapped back to a bacterial species. And so After we documented that snakes indeed have a skin microbiome, we decided to study snake fungal disease and skin microbiomes at the macro scale, which included 5 states in the southeastern United States. The mesoscale, which included 4 distinct Tennessee ecoregions with very unique vegetation, elevational differences, and animal communities. And at the microscale, in 3 of 4 of these distinct Tennessee ecoregions where we had adequate replication. And so when running Permanova analyses to understand differences in the structure of the microbiome, we were able to determine that host species and the interaction with geography and season, and most interestingly to us, statistically significant interaction between host species, seasonality, and the presence or absence of the snake fungal disease pathogen. So all of these are strong predictors of the structure of the snake skin microbiome. Because we're interested in scale and ecology, we then scaled down to the mesoscale, and we saw the very same trends, or most of the same trends, holding true. And at the microscale, once again, the very same trends were holding true. And so when we're observing similar patterns across spatial scales, we can kind of conclude that factors like host snake species, geographic location, season, and pathogen presence are strong predictors of skin assemblages across both broad to fine spatial scales. And so after we observe the presence of the snake fungal disease pathogen and its important and its thought of importance to be like a structural, a factor that structures the snake skin microbiome, we decided to think a little bit more about this idea of pathogen-mediated disturbance to the microbiome. Right? And so initially, our thoughts were kind of rooted in disturbance ecology. And the reason for this is because disturbance ecology allows for us to predict perturbations on biotic communities. And some of the classic examples are clear cutting, drought, and wildfire. However, we don't really call it disturbance in the microbiome world. And in fact, we call it, and have given it a different term, we refer to disturbance to the microbiome as dysbiosis. And there are some very specific trends or patterns that can be captured to describe patterns of dysbiosis, including the loss of beneficial species, gain of pathobionts, loss of microbial diversity, and alterations in microbiome function. And so what I'm going to do is kind of walk you through some of the results that we have generated that point to these different patterns of dysbiosis in the microbiome. Right, and so this dysbiosis we think is pathogen mediated. We know that snake fungal disease causes both chronic and long-term infections to snakes. You can see some of those clinical signs here. Interestingly, the fungus is really cool in its ability to enzymatically alter the skin environment. It produces keratinases and lipases. And it just so happens that snake skin, just like our skin, is made mostly of keratin and lipids. And so these keratinases and lipases directly target those particular carbon compounds. And so we were really degrade them, enzymatically degrade them. So we were really interested in understanding, like, how does the microbiome respond to this fungal pathogen-induced dysbiosis? And so the first major assumption that we really had to answer was, like, can our quantitative PCR assay actually be used as a proxy for disease progression? And so we performed a live animal inoculation experiment on ribbon snakes, and we can see that fungal load does indeed increase over the course of experimental infection in this live animal study. So we confirmed that quantitative PCR is a good measure to predict disease progression, which is really a really important assumption for the rest of what I'm about to show you. So the next thing that we wanted to ask was, does the fungal pathogen load correlate with skin microbiomes across spatial scales? And so what we're seeing here is fungal pathogen load as a blue vector. we're seeing each of the blue points here are representative of a snake skin microbiome in the presence of Ophidiomyces. And each of the pink points is representative of a microbiome in the absence of Ophidiomyces. And so we can see that the microbiome of the snake fungal disease pathogen correlates with fungal pathogen load. And this happens once again across spatial scales, at the macro, meso, and micro scale as well. I'd also like to point out now that this was a rather limited data set. It included only 158 snakeskin microbiomes. And so I'm going to show you in just a few moments about how we've actually expanded on this particular data set. The next logical question that we wanted to ask was, can the microbiome function as an extension of the innate immune system? And so to do this, we went out and caught timber rattlesnakes and black racers. We isolated about 60 different bacterial strains from these two snake species, and then we challenged them against the snake fungal disease pathogen. And so the way that we read these assays, in this Petri plate, we can see the control. This is just a lawn of the fungus spread across the Petri plate. In this case, we have a single bacterial species streaked down the center of the plate, showing no activity. And this is incredibly apparent when looking at the rest of the Petri plates. We can see strong antifungal activity or perhaps competitive exclusion of these different bacterial species when they interact with Ophidiomyces, the snake fungal disease pathogen. So we got pretty excited about this when thinking about innate immunity and the ability of the microbiome to aid in pathogen resistance. So next, what we wanted to do was to go out and perform a live animal inoculation experiment to really more fully understand pathogen-induced dysbiosis. And in this experiment, we used northern water snakes. And so just to give you an idea, these are some of the clinical signs associated with snake fungal disease. They can be really subtle, like a crust on the animal's face, or a bit of a crust on the scale on the ventral side of the animal here, or really severe mycosis. And so this is kind of when we get into some of the heavier data associated with the talk today. What we wanted to look at was alpha diversity of the microbiome, and to measure alpha diversity, we measured species richness, or the number of bacterial taxa on the animal's skin, and Shannon diversity, which takes into account both richness and evenness. On the x-axis, you see experimental time in days. And I should point out that the two left panels, or red points, are experimentally inoculated snakes. And the two panels on the right-hand side, the blue panels, are the sham or control group that didn't receive inoculation. And so what you'll note are these significant and nonlinear trends that were detected for both richness and diversity in the treatment group, but not in the control group. What we also wanted to look at was beta diversity of these microbial communities. And so we did this using three common metrics in this case. We use what's referred to as the Bray-Curtis metric. And so this emphasizes abundant taxa, the Jacquard distance matrix, which emphasizes the presence or absence of bacterial taxa, and the Raup-Crick, which is a null modeling approach. It allows for us to control for differences in richness, differences in alpha diversity between individual samples. So the way that we interpret these plots, we can see once again the inoculated or treatment group in red and the control or sham group in blue. On the y-axis, we see distance to centroid points. And so what this is really a measure of is the amount of heterogeneity or variability in the community. And so what you can see is that snakes that had the fungus were more variable relative to the control for both of these two distance metrics. In the third panel, what we're actually looking at is an increase in heterogeneity over time relative to the control. And so this is interesting. What it indicates is that the snake skin microbiome becomes more variable or more heterogeneous over time as disease progresses. And so what we kind of settled on in our working hypothesis after publishing this paper was that pathogen-induced dysbiosis results in nonlinear trends in bacterial richness, or the number of taxa that are present on the skin microbiome, and an increase in microbiome heterogeneity over the course of disease progression. And we'll come back to those trends in just a bit. And so, as I told you, like the first data set, the first field data set that we have was, it was okay. It was 158 animals, which took a lot of time and a lot of effort to collect. But after developing the snake fungal disease working group, we were like hugely able to expand on our sampling efforts and collect and sequence nearly 800 samples from 31 of the 32 snake species across 5 years in Tennessee. And we were able to cover all 8 of the distinct Tennessee ecoregions. And so we have great spatial diversity as well. And so if I'm putting you to sleep at this point, I thought I'd kind of like spice things up a little bit by showing you how much time and effort really goes into sampling these individuals. So this is one sampling session. You can get a feel for how long it took by just looking into the background and watching the sun set. And if you get bored or are bored right now, keep an eye out for a pineapple and the evolution of this pineapple throughout this video.
Reluctantly crouch at the starting line. Engines pumping and thumping in time. The green light flashes, the flags go up. Churning and burning, they yearn for the cup. They deftly maneuver and muscle parade. Burning fast on an empty tank. Reckless and wild, they pour through the turns. Their prowess is potent and secretly stern. As they speed through the finish, the flags go down. The fans get up and they get out of town. The arena is empty, except for one man still driving and striving as fast as he can. The sun has gone down and the moon has come up. And long ago somebody left to get the cup. But he's driving and striving and hugging the turns and thinking of someone for whom he still burns.
Yeah, one of my undergrads put that together. I love that video. Anytime I need to pick me up, I just watch that. Okay, so what we're going to look at is what we're calling this kind of like improved spatial data set now, and see what types of patterns we uncovered in the microbiome. And so we've also been able to take, since we have so many samples, we've been able to do some really cool species distribution modeling of snake fungal disease in Tennessee. And so you can actually see, and this is work that Matt Grisnick has done and is continuing to work on now in Carolina. But you can see areas that seem to be more or less suitable, warmer colors being more suitable for the fungal pathogen, cooler colors being less suitable. And so there are direct conservation implications for the type of work that we're doing. Okay, and so looking at microbiome diversity in this improved spatial data set, I'd like to point out that, right, because we are going to the landscape, snakes were not repeat sampled. And so we don't have a time series data set like our inoculation experiments. So instead, what we did was to use pathogen load as a proxy for disease progression over time. And I hope what you'll note are some really interesting trends. Once again, nonlinear and consistent loss of OTU richness or bacterial richness in the microbiome, as well as for diversity of the microbiome, Shannon diversity of the microbiome. And so, like, get pretty excited about this. Like, there are some pretty striking similarities in the patterns between live animal inoculation experiments and the field collected results, both indicating that the loss of richness and diversity are due to disease progression. So next, what we wanted to do was to study the structure of the skin microbiome. And what we did was to use something called the Jenks natural breaks algorithm to categorize pathogen load into disease negative animals, low to moderate disease, and severe disease. And we were able to then perform this ordination, this constrained ordination technique to show the average structure of the microbiome represented by these large points. These represent the average of the cloud of points, the individual microbiomes surrounding these centroid points. And we were able to show that the structure of the microbiome does indeed change, and these changes correlate with both richness as well as pathogen load in a unique way. So we also found really striking similarities and patterns of beta diversity between our live animal inoculation experiments. Recall, we've seen an increase in microbiome heterogeneity over time. We saw the very same trend from field-caught animals in our larger expanded data set, which indicated to us that microbiome heterogeneity increases due to disease progression. We've also been working with a computer scientist at MTSU named Josh Phillips and his PhD student, David Ludwig. And what they're actually using are algorithms that are similar to ChatGPT in that they're able to understand the sequence of a DNA, because DNA sequences are structured in a particular way, right? They're structured into a series of codons. But the pool of sequence data that we generate from a high throughput amplicon sequencing run is unstructured in a unique way. And so they've been developing these deep learning models called SetBERT. And these SetBERT models allow for us to predict, to learn the microbiome data, so actually learn the DNA sequences themselves in this particular way, and make predictions about disease state with 99% accuracy, which is just kind of like absolutely astonishing. It blows all of the different modeling approaches that we've used previous to this out of the water. So this is a plot showing our previous modeling approach relative to this newer machine learning or AI-based approach. And so you can see an ordination of in this case, of the microbiomes for disease positive and negative individuals. And the cool thing about these models, and I think like AI-based models in the microbiome world oftentimes get a bad rap because they're oftentimes thought of as like a black box. Like what do we tease out? What can we learn? Like, yeah, we can make a 99% accurate prediction about disease state based on learning the microbiome, but what about that can we actually learn? What can we tease out of this black box? And so this is something unique that Josh Phillips and his group have brought. They're called attention attribution scores. So they allow for us to kind of focus in on particular DNA sequences or particular bacterial taxa that might be important at describing differences between sick snakes and healthy snakes. And so the easiest way, and the way that Josh kind of pitches this idea, is that if you want to accurately identify a picture of a dog, you focus on things like a hairy, furry creature, and you blur all of the stuff out in the background. So it's similar to what we're doing. It's like, what microbial sequences or what microbial taxa are most important, and let's blur out all of the background noise and not pay attention to things like trees and grass, because those don't accurately allow for us to identify the image of the dog or the bacterial taxa that are most predictive of disease state. And so we've been able to predict that species like actinobacteria are really important in describing disease state. So just to give you a feel for it, these are our raw data, our relative abundance data. And if you focus on the actinobacteria, they really don't look all that much different. Their relative abundances are basically the same between disease positive and negative individuals. However, if you tease out the attention attribution scores, or what is allowing for us to make this 99% accurate prediction, it is indeed the actinobacteria that are most predictive of a disease negative state. And this is really cool because within the actinobacteria are species like Streptomyces, which are really well-known secondary metabolite producers that produce all sorts of clinically important antimicrobials. And so like this might actually hint at the importance of the actinobacteria in disease state, predicting disease state, maybe even protecting the snakes from snake fungal disease. And so what I've showed you now are experiments that we've done at the landscape level, some experiments that we've done in controlled live animal studies. And the last thing that I'd like to talk to you a little bit about today are some of these synthetic microbiome experiments that I'm super excited about, because once again, it allows for us to control all abiotic factors. It allows for us to control and mimic host skin chemistry, so we can mimic snake skin chemistry inside of these 96 well plates to understand how Ophidiomyces interacts with bacterial communities or individual species of bacteria. And so we first kind of got put on to this when working on a project with Bob Mason and Charlotte Van Moorlegem, where we were looking at the lipid chemistry of snakes. And so Charlotte was able to collect the snake skin lipids using a mild solvent and impregnate that into a Petri plate-based medium, and then grow Ophidiomyces on the Petri plate. And you can see that Ophidiomyces does indeed grow much slower and is inhibited by the skin lipids relative to the control. And so we talked to Bob and Charlotte and asked them, like, well, what types of skin lipids are present in snakes? And they said, well, the most important and most abundant compounds in snake skin are things like keratin, which is not a lipid, it's a protein, oleic acid, and squalene, as well as cholesterol. So we said, okay, well, let's make a minimal medium. Let's take those carbon sources and put them into a medium and make that the only carbon source that's available to Ophidiomyces. And let's see how it grows to see if we can tease out this interesting effect that we see in the upper left-hand corner. And so we can see, kind of as predicted, Ophidiomyces grows really well on keratin compared to the control, and not in red here. And both oleic acid and squalene actually inhibit the growth of the fungus. And so we've followed up and are currently working on several additional experiments where we're actually looking at these lipids and their concentration-dependent effect on the snake fungal disease pathogen. So what we're seeing here is in blue, the control, and increasing lipid concentration from left to right, with the most concentrated being on the right-hand side for oleic acid. And we're seeing complete growth suppression in a 1% medium. However, this really interesting pattern here, where we actually see that Ophidiomyces is facilitated by very low concentrations of oleic acid. It grows better with very low concentration. So what we're now thinking and talking a little bit about is, like, what's biologically relevant? Like, which one of these bars is actually biologically relevant? And of course, it's not just oleic acid by itself. It's an interaction between oleic acid, keratin, and these other types of skin lipids. And so the next step, we know that keratin is a major component of snake skin. We know that it facilitates snake fungal disease growth. And so we decided to move forward with some synthetic microbiome experiments using keratin as the sole carbon source. Right? And we did this because bacterial fungal interactions are really interesting and relevant when thinking about host chemistry gradients. Right? Bacterial fungal interactions have these different physical associations, from planktonic to really intimate, like intrahyphal colonization. The bacteria and fungi communicate with one another in a variety of different ways, including antibiosis, metabolic exchange, and so forth. And these all result in alterations to the different partners, including changes in community structure, changes in growth, symbiosis, and pathogenicity. And so a PhD rotation student and I designed this experiment called the direct bacterial fungal interaction experiment. And so these are done in 12 well plates. Each of the wells is approximately the size of like a quarter to about a $0.50 in size. And so what we do is pour a solid agar base into the bottom of each well that contains only keratin. We remove the agar plug and replace it with a growing plug of Ophidiomyces. We then place a broth overlay over top of that agar medium and inoculate either one species or a complex community of microorganisms into this well. And then, just like if you've ever done like any cancer cell biology or heard of cancer cell biology studies, we passage this experiment over time. So 6 times, where we aspirate the broth, the spent broth, and collect the microbial community. and then replace it with some fresh keratin broth. And this allows for us to simultaneously quantify how the bacterial community changes in response to the fungal pathogen, as well as how the fungus grows. And we can use a variety of different downstream assays to determine how the fungus grows, its metabolic gene signature, the genes that are expressed, and how the community changes as a whole. So I'll show you some preliminary data associated with this direct bacterial fungal interaction experiment. So this is, there's a lot going on this slide, but I'll walk you all through it. So what we had here are individual bacterial species combined in co-culture in the experiment that I just showed you. So this is one bacterial species and the fungus. The bacteria are Chrysobacterium, Syntobacter, Stenotrophomonas, and Myroides. In the top set of panels, we're seeing fungal growth on the y-axis over time. And in the bottom set of panels, we're seeing bacterial growth in response to either keratin or the control. So the gray dotted line is the fungus growing just by itself. This is a fully factorial design, the fungus growing just by itself. And the red solid lines are showing when the fungus is growing in the presence of the bacterium. We move down to the bottom panel. This is showing, the red box is showing when the bacteria is, or how the bacteria is growing in response to the fungus, or how the bacteria just grows by itself. So there's some really interesting patterns here, variable responses as the slide indicates. So here for a Syntobacter, we see nothing, no interesting trend. It doesn't matter if the bacterium is present, the fungus just doesn't care. Same thing for the bacterium. Bacterium doesn't seem to care much about the fungus or really grow all that well on keratin in general. However, if we look to like Chrysobacterium, we see a really interesting response. In this case, we see Chrysobacterium actually suppressing the growth of the fungus. And Chrysobacterium is facilitated by the fungus. So it grows better, Chrysobacterium grows better in the presence of the fungus, and at the same time inhibits fungal growth. For Stenotrophomonas, we see a somewhat similar pattern. Once again, Ophidiomyces growth is suppressed, but it doesn't really seem to matter. Like the bacterium Stenotrophomonas grows just the same in the presence and the absence of the fungus. So I think there's like a lot left to be unpacked here, especially in the context of doing this in like a community ecology type area where these experiments were only done in monoculture experiments, which doesn't necessarily mimic what the microbiome, like what the microbiome actually looks like. It's much more complex. Okay, the last set of experiments that I want to show you all today are these spent media experiments. And so these spent media experiments allow for us to assess host resource gradients and the production of fungal metabolites. So this is work that we're doing with Justin Miller and Jeff LeBlond at MTSU. And so in theory, if we grow, so this is Ophidiomyces growing in a liquid medium. If we grow this fungus for a longer time period in a keratin-based medium, we would expect for it to use up more of the resource and expect for it to produce more metabolite over time. What we can then do is separate the mycelium using vacuum filtration away from the spent broth and collect just the spent broth. And then we can grow these different bacterial species or communities on the spent broth to determine whether or not there is competitive bacterial fungal interactions or cooperative bacterial fungal interactions of bacterial growth over time. We also further, what we plan to do is actually separate out and just target the remaining carbon that was not metabolized by the fungus, as well as the secondary metabolites that are produced by the fungus to see, like really drill down to the nitty-gritty detail to see exactly what the bacteria is responding to. Is it a carbon or is it metabolite? But some of the results, some of the preliminary results that we have for this experiment are shown here. And so we did this work for 18 different bacterial isolates. You'll see the M9 control, the keratin medium by itself, and then the spent media at 5, 9, and 13 days. And you'll see some really interesting trends here. So 3 of 18 of the isolates that we tested show cooperative bacterial fungal interactions. They simply grow much better in the presence of the fungus than on the keratin medium itself. 9 of 18 of the isolates show cooperative bacterial fungal interactions, but across a response gradient. So they grow really well on 5-day spent media, but that starts to taper off by 9 and 13 days. And then 6 of 18 of the isolates just don't care. In fact, they probably don't grow very well on keratin at all. There's really no bacterial fungal interaction. And so what I'm hoping that I've convinced you of today, as I kind of like wrap things up, is that dysbiosis is clearly occurring in the snakeskin microbiome, but there's a lot left to be studied. I think I've shown you that we've had a loss of beneficial species, a gain of in diversity, a gain in pathobionts, changes in microbiome diversity, both at alpha and beta diversity levels, and alterations in microbiome functions, although I think there are some improvements that we can still do to this 4th point here. And so just to kind of synthesize things, right, this is my general synopsis of fungal-induced dysbiosis on skin microbiomes. What we think is that the fungus starts to colonize snake skin. It utilizes the available niche space, and so the niche space is altered and perhaps shrinks as the fungus continues to grow and be pathogenic. And this results in these nonlinear trends in microbiome diversity. And it's really important to keep space and scale and season in mind, because we know that not all areas across the state of Tennessee are equally suitable for the snake fungal disease pathogen. And then this is the detailed synopsis of what we've learned thus far. So we know that the host supports microbiome assembly. We also know that the host produces lipids that suppress Ophidiomyces growth. The fungal pathogen colonizes the snakes. The fungal pathogen colonizes the host skin. We also know that the fungal pathogen produces metabolites that likely result in variable selection of the microbiome. We know that the microbiome has an innate immune function against, or has innate immune function for the host, and that it produces antifungals or competitively excludes the snake fungal disease pathogen. And the 2 black arrows is what we're placing most of our time and energy into now, really kind of thinking about the types of experiments that allow for us to assess this fungal altered skin chemistry and how niche space changes and how this might result in variable selection on the microbiome, with really placing a strong emphasis on host lipid chemistry, trying to really understand how host lipid chemistry might drive these interactions. And so with that, like, a lot of people and funding sources to thank. You can kind of see that large list here. And if you're interested in what we do in the lab, snap a picture of our QR code and look us up. We're always looking for quality graduate students, both at the master's and PhD level. And so, yeah, I don't know how long I was supposed to go, but if there's time or any questions, yeah, I'm happy to field those now. Or if you want to talk about anything that's not really research related, my path to where I am now from ESF to now, that's fine too. Whatever y'all want to chat about is good by me. Oh, Dr. Weir. Oh, goodness. I'm glad I can't hear you.
I'm sorry I missed the first 15 minutes or so of your talk there, but very interesting.
I'm glad I didn't see. I would have been much more nervous.
That's why I kept the light off behind me. I thought I'll hide in the shadows.
Yeah.
I did have one question, actually. I may have missed this. It might have been something you talked about early on.
Mm-hmm.
Are there any patterns in terms of infection of snakes and the age of the snake?
Mm.
Like, are young snakes free from this disease?
I think not.
No?
No, I think this pathogen, or at least what's hypothesized, is that it oftentimes overwinters with the animals as they go into hibernation, and
Yeah.
these animals all emerge with what was called like historically as like hibernation blisters or sores. And when they emerge, some of them are able to kind of undergo like several rounds of skin shedding and rid the pathogen,
Yep.
and they're just fine, but others succumb to disease. So I think this pathogen loves keratin and maybe a combination of the different lipids that are there. And I don't think that it really cares all that much about how old the animal is or anything like that. But I think something to be tested.
And are there any Tennessee snakes that you've never found it on?
We've only not sampled one species,
I think I caught
and
that.
yeah, and we have found it on all of the others that we have sampled. So, but it does seem to be more prevalent on water snakes for the most part, and it could just be like that aquatic habitat constantly being wet, perhaps. Could also be a product of we sample a lot more water snakes because they're easier to catch. Yeah, I think still a lot to be uncovered and associated with this disease and who's most affected.
Yep. Well, very, very interesting.
Thank you. So before you go, I still have my I can't see my camera. Can you see this? Hold on. Let me turn my camera on. So anytime I'm all stressed out,
Oh my goodness, look at that.
I still have it. I still have the slide collection that goes with it too. It sits right next to me on my shelf here.
I
Anytime
hope
I'm
you're all listening to
stressed
that.
out and cursing out all the paperwork that I have to do, and yeah, I look at that and I'm like, "Ah, it's worth it. This is all worth it."
Well, that'll be a story I can tell next year's group. You don't need to get rid of this thing. Excellent. Anybody else got any questions for Donny?
Jeremiah, did I see you raise your hand earlier?
Yeah, don't be shy.
I got a question if no one else does.
Mhm.
Just clapping, okay. Um, I guess how is it well known how the snake skin microbiome is established? Like is it mostly coming from the environment or some of it passed down?
It is um, not known. Some snakes are live born, other
Mhm.
snakes are not. Um, and so I can imagine that there's probably some maternal effect there. Some snakes after they're born will hang out together, kind of like almost like pile on each other and spend time together for probably the first several months of their lives. Like we've documented copperhead babies all kind of piled in their mother's nest, just laying on top of one another. So I can imagine that there's probably some unique and interesting maternal effect. So vertical transfer relative to horizontal between individuals and from the environment to those individuals. I think it'd be an interesting question to ask. Although I think that if you're excited about maternal transfer, vertical or horizontal transmission of the microbiome, I think there are probably better systems to ask it in than snakes.
Hmm.
Yeah,
Cool.
we have another data set right now that is striking. It's a wood frog gut microbiome data set, and the maternal effect on these animals is unbelievably strong. It's an interaction between gut fungi and the mom, but the maternal effect really stands out. And then the trend is exactly reversed for the gut microbiome community, the fungal community that's living there. So mom influences the bacterial community, but a fungus called Ophidiomyces seems to co-structure and interact with the bacterial community, but mom is the main driving force. And for the gut fungal community, mom seems to be a secondary factor, and it's really the Ophidiomyces that seems to be the main structuring element of the gut microbiome. So I think there's better systems if you're interested in microbiome transfer and maternal effects than snakes.
Yep. The maternal effect definitely sounds very interesting with the vertical transfer. Thank you.
Mm-hmm.
All right.
See similarities in snakes and amphibian. And amphibian, Aiden? Yeah. Yeah, so we have pretty extensive data sets now from snake skins, salamander skins, frog, as well as gut microbiome communities from those very same groups. And host is a major structuring factor. It seems as if host is like one of the most predictive at determining the structure of the gut bacterial microbiome. The gut fungal microbiome, I think we're still doing some sequencing to really determine that, but host group is important. Geography seems to be secondary, but host seems to be most important. You're welcome.
All right. Well, I'll give people a couple more seconds to think of questions, but in the meantime, thanks, Dr. Walker. I really appreciate it. That was great, doing a lot of really interesting research down there. Also, thank you to the Herpetology Club. Thanks, guys, for coming out. We appreciate you taking the time. For ESF Mycology Club members, we hope to see you guys this Friday for a bi-weekly meeting in Baker 148 at 6 p.m. The topic is going to be mycoheterotrophs, or plants that parasitize fungi. It's going to be a pretty interesting discussion, so we hope to see you there. And thanks for coming out tonight.
Thank you so much for the invitation. It's great seeing you again.
Thank you.
Donny,
Yeah.
great to see you.
Yeah.